Okay. So this lab is on the topic of osmosis and dialysis. So we have sort of a general concept of what, well, we have a definite concept of what osmosis is based on what we studied so far. Dialysis is sort of a subset of osmosis, and it's designed, all right. Osmosis, let's see, I want to be sure I'm consistent with their definition. Yes. Osmosis is the movement of water molecules through a semi-permeable membrane. And the driving force is a difference in concentration on either side of the membrane. So the key here are two things: water and semi-permeable. Semi-permeable membranes only pass water and molecules that are smaller than water. Gas molecules will go through a semi-permeable, no problem. And what we're trying to do here is simulate cell walls, cell membranes. So which way does the water move? Well, it moves from the higher concentration of water to the lower concentration of water, which means it moves from the lower concentration of solute to the higher concentration of solute. So it moves in the direction of the hypertonic solution. All right. Contrast that with dialysis. Actually, dialysis is a special type of process that's used primarily for people that have failing kidneys. But in that case, it's called, oh yeah, okay, dialysis. This involves the movement of selective size molecules. These go through a selectively permeable membrane. And this is also based upon a difference in concentration. Now we're talking about a difference in concentration of those molecules of different sizes. Okay, so the example we're going to use for osmosis today is the potato. Because it's just chock full of cells. So you take a potato core and you submerge it in a solution that is hypertonic, pure water, and the water will move into the cells of the potato core and make it get rigid, hard as the cells swell. And it'll also make it get longer. You're going to measure that. Let's see. If we soak it in isotonic solution, you should see relatively little difference. Right? If you soak it in a hypertonic solution, the water is going to move out of the potato into the hypertonic solution. And the potato should shrink and get limp. Right? Remember, everybody's had biology courses, studied turgor. Understand that word, turgor?
I know it's about.
Oh, well, turgor is an expression of stiffness, and it's in response to hydraulic pressure inside the cells. So if you get more pressure inside the cell, it gets stiff, turgid. If you suck the water out of it, then it gets limp, less turgid. Okay, so the potato, we're going to soak in pure water in 0.9% sodium chloride and 20% sodium chloride. Those are our three. And we'll talk about mixing those solutions in just a minute. You have to make your own solutions. Then we're going to do dialysis. And our membrane for dialysis is, maybe when this happens. Look it up. Save time and just look it up. Looking for the right word. Our semi-permeable membrane, oh, duh. It's called dialysis tubing. So the dialysis tubing is, I'm preparing it right now. You have to soak it in distilled water to prepare the membrane. So it's ready to go. And this tubing, it's flat right now. So you should just put it between your fingers and move them back and forth, kind of like opening a plastic bag. And you can find the end of it where it's open, and then you like stick a probe down it and just make sure it's open all the way down. And then you tie a knot in one end, and then you follow the instructions and fill it with solution that you make up. And then you tie off the other end with a string. So your one partner will hold it up, and then you'll wrap a string around it and tie it off. So nothing gets in through the open ends. It has to go through the membrane.
I thought you were talking about something really, really, really small. Like you have to tie something really small. I didn't realize it was going to be big.
Yeah,
That's why I
well,
was like, how do you do that?
the tubing, once you open it up, it'll be a little bit bigger around than this marker, and about that long. Okay. So this is going to simulate kidneys or a dialysis machine at what do I call them? What are those labs called where people go with kidney failure? Dialysis lab. Yeah. And they sit there and they have their blood, they've washed their blood. The blood goes through these types of tubings, and the solution on the outside of the tubing is hypotonic to these molecules, right? Like urea. You got to get that urea out of your blood because the kidneys normally do that, and some other contaminants. And then they run the blood through there and they give it back to you. And you have to do that, some people 3 times a week, because it builds up so fast. But the differential here, the real key to the dialysis tubing is molecular size. And we're going to use 2 different molecules. Dextrose, or we could say glucose. Same thing for our purposes, same size molecules, as compared to starch. Right? Starch is glucose in a long chain, big polymer. So that means this is the small molecule and this is the big one. So what would we expect from this? Well, if you put both of them inside that tubing and then immerse it in distilled water, which one's going to move through the tubing? That one we expect. And this one's going to stay behind. So our testing is you're going to test the solution inside the tubing and the solution outside the tubing, I think. Let's see. No, just outside. We don't need to test inside because we know it's already there, because we put it there. So you just test the solution outside the tubing. See which one moved through the tubing. And starch would test with iodine. That's a really simple test. It's turned dark blue or black if the concentration is high enough. And we have a solution. We have the test for glucose is Benedict solution. It's kind of a bluish color because it has copper in it. Only you have to heat this one up when you add them together. It's a qualitative test. And you test for glucose, and you have to heat it up in order to develop the color. So those are the two tests. But we need to know, we need to have a standard, right? So first you're going to do tests with pure starch and with pure glucose. This with iodine and this with Benedict's, just to know what it looks like. And then you're going to go in and test the solution that's outside the tubing. So what else? That's basically it. So how do you heat up this Benedict solution to make it react? Hot water bath. Right, so you're going to have a little beaker. It doesn't take a big beaker, like a small beaker, 100 milliliter beaker with water in it. So it should heat up pretty quick. Oh, okay. So pre-lab question has to do with how do you prepare your solutions of salt solutions. Right. I know why they put these things back. Pre-lab questions are the last ones. That's so that you can fill them out and rip them off and hand them in. We're just going through a pre-lab discussion. Okay, the first question is, calculate the amount of sodium chloride needed to prepare 50 milliliters of 0.9% sodium chloride solution and 20% sodium chloride solution. So the key is, what do we mean by 0.9%? I've said it before, percent is a dimensionless number. I mean, that's a dimensionless unit. It doesn't mean anything unless you know what it was derived from. This is mass volume. Mass volume. Now we know something. So this will be parts per 100 means 1 part mass and 100 parts volume. And what's the relationship between mass and volume? Well, water is 1 gram per milliliter, so 1 gram equals 1 milliliter. So mass volume would be 0.9 grams per 100 milliliters. Okay? Now that we know that, we can calculate, let's see, what did it say? 50 milliliters. We're going to make 50 milliliters of solution. So how many grams of sodium chloride do you need? 0.45, right? Half. I mean, we could go out and do this. Right? If you're going to have 50 milliliters, let's see, how would we do that? So it's times 2. Oh, no, it's the other way around. Excuse me. Right. If we're going, actually, that's not the way to do it. That's even confusing me. The ratio is the important thing. 0.9 grams per 100 milliliters is the same thing as 0.45 grams per 50 milliliters. Right. They're directly proportional. If this one goes down, that one's got to go down too. So this one goes down by half. This one goes down by half. 0.45 grams in 50 milliliters. And for convenience sake, it only has to be approximate. So instead of saying dissolve 0.45 grams in 50 milliliters of water in a graduated cylinder, just put 4.45 grams in your container and add 50 milliliters of water. That's close enough. How about the other one? 20%. Right. That would be 20 grams per 100 milliliters. So in 50 milliliters, how many are you going to need? 10 grams. That's a lot of salt. It'll take a while for that to dissolve. So it was me, I'd weigh this one out first and get it started. And go weigh out the other one. All right, so that takes care of pre-lab question 1. You can do 2 by yourself: isotonic, hypotonic, hypertonic definitions. Let's see. What's the function of dialysis tubing? Well, in practical use, dialysis tubing well, let's see. Usually these pre-lab questions are derived from the method itself. Purpose of dialysis tubing. Yeah, I missed my guess. I don't see a purpose for dialysis tubing. Oh, they're using a word in here. I just noticed this. They're using a word that we haven't talked about. So when you put your tube in here, it's tied off there and it's tied off here, and it's in this solution. This solution outside the tube is called the dialysate. Okay, and in our case, it's pure water. Distilled water. So the purpose of dialysis tubing is to, I guess you could phrase it a number of different ways. From a chemist standpoint, the purpose of dialysis tubing is to limit the flow of molecules between solutions based upon particle size, based on molecular size. But dialysis tubing used in the hemodialysis lab is to remove certain molecules from the patient's blood based on size. Actually, we're basing this on size alone, but dialysis tubing comes in a whole host of molecular structures, and it can be very selective, not just on size, but on polarity of the molecules. Or you can look it up. Just regurgitate the answer from the internet. Let's see, what else is here? What's the purpose of the Benedict solution? Is to detect the presence of glucose in your solution. Does it ask about iodine also? Yeah, what's the purpose of iodine? To detect the presence of starch. Okay, let's see. Here we go. All right, we've got tables to fill out. Don't try to do the method based on the table. I mean, follow the instructions. The table is just to collect your data. Okay, post-lab questions, I'll let you deal with that. Let's see. Yeah, there are your tables. So we got, looks like 3 of them, right? 3 tables. The first table is potatoes. So you're going to measure the mass of the potato core, the length of the core in millimeters. So you need a ruler. Millimeters. Something like this. Each of your kids should have one of those. And the texture. Just say, right, you know, is it rigid? Is it limp? Is it squish? Things like that. That's before, and then after, do the same measurements after it's soaked. The dialysis test the tables are separated into Benedict versus in Benedict solution in one and iodine in the second one. So, for instance, the first measurement here, Benedict with distilled water, negative control. So you're going to add the Benedict to it and heat it up and say, all right, what does it look like? It might have a color to it because the Benedict is blue. And then you're going to add glucose. Do it the same thing with glucose and test that and see what the color is. And then you're going to test the dialysate. Oh, it does test the Benedicts with the solution inside the tubing. Okay, so you're going to do 4 tests. You're going to give a solution before it heats, solution after it heats, and then you're going to say, is glucose present? So that's just a checkbox, whether it's present or absent. And the same thing with iodine, except iodine you don't have to heat, so it's a simpler test. Okay, any questions? All right.
Yeah, not totally. Not fully.
Oh, yeah, okay. You reserve the right to recall the witness.